And what about that genome information? DNA is not a blueprint. You can't, if you look at DNA and you try to, they talk about decoding the genome. They can't decode the genome. They're just cataloging the genome. That's what they can do, catalog the genome. You can see, oh yeah, there's a ACTG, 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 those amino acids. And if you try to read the DNA, you'll never figure out what the organism is. But the DNA is actually coded information. It's coded information. It's not just random mutations and random collections. It's coded. And that those codes specify how the organism develops from initial, if it's embryonic, or how it divides if it's single cell, and carries the instructions for ongoing operations. It's all right there. But it's only information. The coded information in DNA is only faintly understood by science. Faintly. The word faintly, I'm going to say, is if you went outside and it was totally black and you saw a little teeny light off in the horizon and you'd say, I think there's a light out there. That's how much information we know. And we have mapped the genome and do bioengineering on a day-by-day basis in science and in industry. Okay? But it's only faintly known because we don't really understand what the coded information is. Most of what we learn is by trial and error and mistakes. DNA is precisely analogous to software programming. It is exactly like a software program. I know this because that's my field, information technology. It is exactly like a software program. And just like a program, if I give you a program and I say, here, use this, what will you tell me? I don't have a computer to put it on. Well, you need a computer too. And that computer is called the cell. The cell is the apparatus that translates the information in the DNA into action. Now, the building blocks of computer hardware and software are very, very simple. It's very simple, like yes, no, that's binary, right? Yes, no, 1, 0. And current flows, current doesn't flow. 5 volts, 0 volts. Very simple things. But the aggregate of that takes intelligence. And it's incredibly complex. Even this PC is incredibly complex. I'll bet most of the people in this room have no clue about it. And all the people in this room don't really understand how the processor works. Okay? At the molecular level. But it's always a result of intelligent design. And all coded information everywhere, anytime you've ever seen it, a message written in the sand, language, writing, design, architecture, art, mathematics, communication, software, is always the result of intelligent agency. And this DNA in the cell, the program in the computer, is evidence for a level of sophistication and intelligence beyond our wildest comprehension. And IT experts will tell you that. Information experts will tell you that. Evolutionists will say it just happened by chance. Because remember, that's their religion. Embryonic development. Okay, it's mind-boggling and unbelievably complex. And it develops, the zygote starts out and develops from a coded instructions in its DNA using its own information processing system. And the development precisely is controlled by the sequencing in the program. And there are control genes, like the Hox gene and the Plas7 gene. And these genes control features and forms that are created from that original zygote. They say, okay, now it's time to turn the zygote into a ball. And when the ball is achieved, then there's a chemical signal sent to all the cells, and another control gene kicks in. And it says, okay, now it's time to start the central mass inside the blastula. And when the central mass creates a certain level, then a chemical signal is sent out, and the blastula gets bigger, and the central mass then starts to differentiate into tissues. And when it starts to differentiate at a certain level, it says, okay, now it's time to start forming a bilateral symmetry. So cells start to move up and down. There's a program running in each one of those cells that knows exactly what's supposed to happen. And these control genes turn off and turn on genes in other cells, in and among themselves, so that the chemical agents going back and forth say, okay, these cells here, you're going to be the central part of the organism. As an example, this is all choreographed. And all embryos across species form along a similar process, using similar control genes. You could say that that's a theory for evolution, right? That's proof of evolution. But the great puzzle is that the evolutionists know the similar control genes, like the Hox gene in me and the Hox gene in a fruit fly, start off different processes, kick off different embryonic processes. So so that you can't take the Hox gene is common across all these genomes, but in each genome, when you start off an embryo, it'll do something different. Not simple local rules.
The beginning of the development of a human embryo begins in the ovary. Here is an unfertilized egg drifting towards the fallopian tube. It begins the fertilization process as it encounters sperm. While many may hit the egg, only one manages to deliver its genetic payload. And when it does, the egg immediately stiffens its outer walls to prevent other entry.
Okay, so control gene took over.
Over a period of about 5 days, the egg divides and divides again until there are exactly 32 cells, now squeezed inside the same stiffened outer wall.
Another control gene kicks in. So
inside the blastocyst, we find a group of cells that will actually develop into a human being. The outer cells will give rise to the placenta and other supporting tissues. At this stage, it implants itself into the wall of the uterus, and the pregnancy is initiated. Now we see the inner cell mass form into a disc.
University
in 14 days, we can see the primitive streak form, which establishes the line of bilateral symmetry. Then the cells in the disc migrate as development continues.
Control genes are turning on and turning off.
As the cells on one side of the disc grow faster than those on the other side, the whole disc rolls into nested tubes that will become the gut canal and other tissues. At this point, the embryo is about 28 days old and measures about 1/4 inch long. You can see all 4 limb buds and early inner ear development. 4 days later, eye pits are forming and the arm buds begin to taper. Another day, and we see the beginning of nasal pits and hand plates. This embryo is now a little less than 1/2 inch long. At 37 days in, it is 3/4 inches long. Nose and ears continue to develop, and thigh, leg, and foot can be distinguished.
These are MRI images.
And we can see fingers, a jaw, and a lumbar curvature. Three more days, and we have elbows and the beginning of eyelids. At 47 days, our embryo has straightened its trunk. Its limbs have extended forward, and toes are visible. Another few days, and the embryo has upper limbs slightly bent at the elbows, short stubby fingers, hands curving over towards each other, but still far apart. 52 days old and an inch long. And we see hand pads, wrists, and distinct toes. 2 more days, and we see external ears, and opposite hands can now touch. At 8 weeks, the embryo is still only 1 1/4 inch long. But the detail is amazing.
Pretty interesting.
The beginning of the
Let's get there we go. There's a law of facilitated variation. The species require a robust structure, regulatory system with a built-in capacity for variation, and a signaling system. That's what we just talked about. And remember, this is all on the internet, so look at it. And then there's one more thing, the law of inverse causality. Cause and effects, science is based on it. Law is based on it. And then B is the result of A, and C is the result of B. It's a direct chain of evidence or causation, but it's not applicable for origin of life because this chain of events never creates life. Like the primitive atmosphere, chemicals, never been able to show any sort of life starting. But biologists say that there is the inverse law of causality, law of inverse causality rules in biology where the end result is the beginning. Okay? The fully informed adult is the starting port for the zygote. The zygote will never be affected by the problems that the adult faces, but must have knowledge of the conditions the adult form will experience to grow. And all these future events must have been in full view of the progenitor's development program, plus the zygote's own development program. And since all this must be present at the beginning for any species to survive, the only alternative is intelligent design. This is starting to grow inside biology with all the biological changes and science that's being understood these days. It's called the law of inverse causality, and it kind of screams there was a purpose. This actually, evolution is no purpose, just random. But biology doesn't work that way. If you take a look at blood stem cells, there's a stem cell, an obvious various control genes as it grows into all these various kinds of different kinds of blood cells. And there is, for instance, leukocytes.